Welcome to the Satellite Symposium of GenDx. The title is Excellence in Sharing. My name is Wietse Mulder, I'm the CEO and co-founder of GenDx, and I'm the chairman of this session. We want to thank the organizers for organizing this virtual EFI conference. We really appreciate what you've undertaken to make this meeting happen, and we hope you enjoy this session. We have an interesting lineup of speakers. The first speaker will be Professor Symposium Bahram, and he'll talk about the role of MIC-A and MIC-B in transplantation. Lisette van der Corpert will continue, and she will talk about the clinical relevance of molecular markers in hemato-oncology. Thereafter, Pascal van der Welen will update you on chimerism, monitoring by next-generation sequencing. And at last, Maarten Penning, he will update on product development of GenDx. This is a pre-recorded session. If you have any questions during the presentations, please write them down, come to our virtual booth and chat with one of my colleagues, and they can probably answer your question. The first speaker is Professor Symposium Bahram. He's Professor of Medicine at the Strasbourg University School of Medicine and Teaching Hospitals in France. And he will talk about the role of MIC-A and MIC-B in transplantation. For now, sit back and relax and enjoy the session.
Okay, very good day to everyone in this virtual meeting, and I would like, first of all, to thank the organizers for giving me the opportunity to make a quite brief presentation on MIC-A and MIC-B histocompatibility genes, and I will hopefully show you their relevance in clinical practice. So this audience is quite acutely aware of the organization of the MHC, and as you see in the bottom of this slide, MIC-A is a non-conventional MHC class 1 molecule, which is actually the closest gene next to HLA-B, which is familiar to everyone. And next to MIC-A, there is a second locus, MIC-B, which we will be talking about today. So these are within the class 1 region of the major histocompatibility complex. Again, very shortly, as you know, MIC-A and MIC-B are ligands for NKG2D. So MIC-A and MIC-B, the MIC gene family, were discovered by us almost a quarter century ago. And these are non-conventional MHC class 1 molecules in the sense, and you will see why, they are highly glycosylated, stress-induced cell surface protein. As you see on the right side, the structure is very similar to structure of a conventional MHC class 1 molecules, in the sense that there are 3 extracellular domains, alpha 1, alpha 2, and alpha 3, transmembrane and cytoplasmic tail. One particularity is that the molecule does not need beta-2 microglobulin for surface expression, neither does it need any peptides, so it's a TAP-dependent, beta-2 microglobulin independent, so TAP beta-2 microglobulin independent surface expression. And the molecule does not interact with the T-cell receptor, but with another important activatory receptor, which is on the surface of certain NK cells and T lymphocytes, especially gamma delta T cells and CD8 alpha beta T cells. Again, our knowledge of the pathophysiology and biology of MIC-A and MIC-B and their receptor is growing by the day. Now we know that these molecules are not expressed at the cell surface normally. You see this on the left of this slide. However, upon cell stress, this could be infection, tumorization, or other kind of stress. MIC-A and MIC-B come to the surface, and once there, they are recognized by NKG2D. However, this recognition this recognition does not happen in certain circumstances, in pathological circumstances, and so there are several ways that the immune system can fail to recognize these molecules, and one of them is by solubilization of both MIC-A and MIC-B proteins. So something which will be of interest to this community, of course, is the polymorphism of these molecules. So as you well know, HLA genes are extremely polymorphic. I think in total there are, as of today, there are close to 20,000 alleles for HLA class 1 and HLA class 2 molecules. However, the so-called non-classical HLA class 1 molecules like HLA E, F, and G have a very low degree of polymorphism, and all other non-conventional MHC class 1 molecules are either not polymorphic or have a very low degree of polymorphism, like MR1, CD1, etc. MIC-A and MIC-B are very different in the aspect that they are quite polymorphic. So as of today, there are almost the same number of alleles for MIC-A and MIC-B, 224, so over 200 for each. And a peculiarity of these alleles is that, first of all, as you might imagine, these are located, the polymorphic positions are located in distinct locations with respect to polymorphic positions of MHC, conventional MHC class 1 molecules. And the other fact is that all three domains, alpha 1, alpha 2, alpha 3, seem to be more or less polymorphic, unlike HLA class 1 molecules, where the alpha 3 domain has very few polymorphic variants. This will show you what is known of the allele distribution in different populations. On the left is our own internal data. We have sequenced up to 10,000 individuals for MIC-A and MIC-B. And on the right is the published data. Of course, they are very similar. So the take-home message is that one MIC-A allele, which is called MIC-A008, is the most polymorphic, is the most frequent, allele within the population, and let's say up to 35 to 40% of wide variety of human populations have this allele. This is, if you will, equivalent to HLA-A2 in the HLA system, HLA-A2 being the most frequent HLA-A allele, MIC-A008 being the most frequent MIC-A allele. However, MIC-A008 is not just another allele, it's a very peculiar allele in the fact that it has a frame shift mutation in its transmembrane segment and has no cytoplasmic tail. And the cytoplasmic tail of MIC-A being known to interact with the protein from cytomegalovirus, which sequesters the protein inside the cell. This is clearly, there has been a selection event for this allele to become so frequent, and it's this allows basically the allele to escape from interference by cytomegalovirus. So I think it's a pretty elegant way to show the influence of population genetics on biology. The situation for MIC-B, again, we have sequenced around 10,000 individuals, and the most frequent MIC-B allele is MIC-B005. So just to finish with the polymorphism at this point, just bear in mind that the 20,000 HLA alleles have been identified upon sequencing of several million individuals, especially within bone marrow registries. MIC-A and MIC-B alleles have been identified thus far by having sequenced only thousands of individuals. I'm not saying that once we sequence millions of individuals, we reach the level of diversity of HLA, but I think there is still room for novel MIC-A and MIC-B alleles to be identified. So I will now quickly show you why we have started the work in order to show the relevance of MIC-A and MIC-B in transplantation. And today I will only talk about bone marrow transplantation. So this is a classical data gathered by John Hansen many years ago, which remains valid. And as you know, in hematopoietic stem cell transplantation, the more you have mismatches between donors and recipient, the higher is the probability of acute graft versus host disease. Once you have total mismatch, you almost have 100% chance of having an acute GVHD. Yet, once you have total HLA genotypic match, so this means donor recipients are share both haplotypes and within the same family, you still have a large number of graft versus host disease. This means that there are histocompatibility loci outside the MHC. And here, the difference between full genotypic match and zero locus mismatch means that there are other genes, this time inside the MHC, which are histocompatibility genes. And for us, MIC-A and MIC-B were the most potent candidates for this. So since many years, we have been doing this work, and it has come to fruition within the last few years. So in a collaborative French and Dutch investigation, we were able to show quite clearly that matching for MIC-A significantly reduced the incidence of acute and chronic GVHD. So, you see here the data which were published in Blood a few years back, that MIC-A mismatch individuals have a higher incidence of acute GVHD, severe GVHD, grades 3 and 4, as well as chronic GVHD. So this has been very clear. And you also see that biologically, the expression of MIC-A and MIC-B is higher in the intestine of GVHD patients versus controls. And as you know, intestine is one of the clear sites for GVHD biology. MIC-B is also involved in GVHD, and we went after MIC-B again through the same collaborative network. And I can't cite by name all the collaborators, but you see them in publications. And of course, without them, this work would not have been happening. And with MIC-B, what is interesting is that cytomegalovirus produces a specific protein, UL16, which blocks basically recognition of NKG2D, of MIC-A by NKG2D. And this has been known. Has been known, and it's worked out at crystal level structure. And we realized that among the key residues which interact, which are responsible for this interaction, there is a polymorphic residue within MIC-B, which is MIC-B position 98. And we intended to see if this diversity had effect on graft versus host disease and in CMV infection in these patients. And to make a long story short, it does. So MIC-B 98 mismatched patients do have a higher incidence of acute and chronic GVHD, and as well as the mismatches are associated with a higher incidence of CMV infection reactivation. And as you know, CMV infection reactivation is a major clinical issue, as is graft versus host disease. So Without making any further ado, I think my time is up. I think this was a short presentation, yet I hope that it was of interest to you and of interest to the fact that including MIC-A and MIC-B testing in patient selection, donor selection, as well as a prognostic factor in GVHD could be of real clinical use. And yeah, having said that, thank you very much for your attention and hope to see you all next time in person.
Thank you, Siamak, for this interesting presentation about MIC-A and MIC-B. And we will continue with a presentation by Lisette van der Korput. She is medical immunologist at the UMC Utrecht in the Netherlands. And she will talk about the clinical relevance of molecular markers in hemato-oncology.
Okay, thank you for the invitation. I'm happy to have the opportunity to present the clinical relevance of molecular markers in hemato-oncology. And I want to present to you that molecular markers are very important for diagnosis, for risk classification, prognosis, measurable residual disease or MRD analysis, treatment and clinical or therapeutical intervention. And to start with, it's the hematopoiesis in the bone marrow, as you all know. And it starts with the hematopoietic stem cells, which divide in common myeloid progenitors and lymphoid progenitors. But for this presentation, I will focus on the myeloid cell lineages. At the left here, the thrombopoiesis with the presence of megakaryocytes producing the platelets, the erythropoiesis producing the red blood cells, and the granulopoiesis with the different types of granulocytes, and the monocytopoiesis for the monocytes. And in every step of these maturation, something can go wrong, leading to an increase or a decrease of certain cell types. So to start with the most common myeloid disorders, the myeloproliferative neoplasms, which can be divided into BCR-ABL negative and BCR-ABL positive, like the chronic myeloid leukemia, the CML, which is BCR-ABL positive, the BCR-ABL negative ones, like chronic neutrophilic leukemia, chronic eosinophilic leukemia, but the most common MPNs are the polycythemia vera, the PV, resulting in an increase of red blood cells, essential thrombocythemia or ET, leading to an increase of platelets and resulting in clinical problems like a higher risk of thrombosis and lightheadedness, and the primary myelofibrosis or PMF. But also patients with PV or ET can transform into myelofibrosis, the secondary myelofibrosis. And this is all the result of the constitutive activation of the JAK-STAT signaling pathway. And around 80 to 90% of the patients show one of the driver mutations in the gene JAK2, CALR, or MPL. And as you can see here, almost all PV patients show the JAK2 mutation in exon 14, which is a hotspot mutation. But this JAK2 mutation can also be present in ET or PMF patients, around 55 to 65% of the cases. Then you can have a mutation in CALR in exon 9 and also present for around 20 to 25% of ET and PMF. The mutation in exon 10 of MPL can also be present in ET or PMF around 5 to 10% of the patients. And the presence of one of these driver mutations forms one of the major criteria for the diagnosis of these MPNs. Patients can also be triple negative, and then it's very important to look for additional mutations, and especially for the high molecular risk mutations. You see here overall survival and risk to transform to acute leukemia in almost 500 patients with myelofibrosis. And when you have one of these high molecular risk mutations in ASXL1, CBL, EZH2, IDH1, IDH2, NRAS or KRAS, SRSF2, U2AF1 or TP53, then you see that you have an adverse risk of the survival is shortened. And if you have a mutation in TP53, you see that the overall survival is even shorter. And when you have other mutations in other myeloid genes, then the survival is much higher. Also, it's important for the risk to transform to acute leukemia. You see that with TP53 mutations, this is higher. Then in the mutation-enhanced international prognostic scoring system plus karyotype, these mutations are now also part of the prognostic scoring system, in addition to clinical risk factors like anemia, circulating blast, and constitutional symptoms like night sweats and weight loss. Also karyotype, which can be unfavorable, and having 2 or more of these high molecular risk mutations, you get 3 points extra. If you have 1 high molecular risk mutation, 2 points, and if you don't have the type 1 like CALR mutation, extra 2 points. And around 8 to 9% of these PMF patients have 2 or more high molecular risk mutations. Then you see here the overall survival curves of the 5 different risk groups with a very good risk and a very adverse risk, bad prognosis. And it's important to know which risk group your patient is in, because then you have to evaluate whether the patient needs to be treated with an allogeneic transplant. Another disorder is the myelodysplastic syndrome, or MDS, which is also characterized by clonal hematopoiesis, cytopenias, and an abnormal cellular maturation. And you see here that in more than 900 patients, they looked for gene mutations in 104 genes, and it was found by next-generation sequencing that 47 genes showed mutations. And the most frequently mutated gene is the TET2 gene, which is not associated with a bad prognosis. The SF3B1 gene mutations, which is associated with a good prognosis. The ASXL1, SRSF2, DNMT3A, and RUNX1, which are all associated with a bad prognosis. And it's very important for these prognostic scoring system, because the additional mutations are not involved in this scoring system, and especially for patients, MDS patients with a low risk or an intermediate risk, is very important to see whether there are additional high molecular risk mutations in TP53, EZH2, RUNX1 or ASLX1 or ETV6, because what you see that the overall survival is shortened when one of these genes is mutated. And so then you can evaluate whether the patient needs to be treated with an allogeneic stem cell transplantation. Acute myeloid leukemia, you see here the bone marrow of a healthy individual with all different type of hematopoietic stem cells. And here leukemic cells, the blasts present in the bone marrow. The AML is a very heterogeneous disease. And you see here different molecular markers with fusion genes that can be present, and also different somatic mutations that can be present. And one of the mutations is in the gene NPM1. Around 30% of the patients is NPM1 mutated. And this is very important to know because this is a very good molecular marker which is used for MRD analysis. It's also important for the risk stratification. This is published in 2017 by the European Leukemia Net. 3 risk groups: favorable, intermediate, and adverse risk group. And if you are in the favorable group, you can have a biallelic mutation in the gene SEPPA, also mutation in the NPM1, and a very low variant allele frequency in the FLT3. Intermediate risk, then you have some cytogenetic abnormalities, and when you are in the adverse risk group, then you can have mutations in the RUNX1, ASXL1 or TP53, and when you have an adverse risk, you need to be transplanted with an allogeneic donor. So here you see the overall survival curves of the 3 different risk groups. It's also important to know whether there are mutations in the gene FLT3, IDH1 or IDH2, because nowadays the patients can be treated in addition to chemotherapy with specific inhibitors. And as you can see here, the overall survival curve is somewhat better when the patient is treated with a specific FLT3 inhibitor in addition to chemotherapy. Then after 2 rounds of chemotherapy, when the patient is NPM1 positive, the MRD analysis is performed to see whether the patient is negative or below -10 to the 4th. And if this is an intermediate risk patient, these patients can be treated with an autologous stem cell transplantation. But if the NPM1 is positive or above the 10 to the -4th level, then this patient needs to be treated with an allogeneic stem cell transplant. It's also important after transplantation, then the NPM1 can be measured. And if it's positive again, then you have to do a therapeutical intervention by giving the patient a donor lymphocyte infusion. So I hope that it was clear that measuring or by next generation sequencing, do the gene mutation analysis is important for the diagnosis, prognosis, risk classification, MRD analysis, so that you can treat the patient with a hematologic malignancy in an optimal way. Thank you for your attention.
Thank you, Lisette, for this interesting presentation. And we will continue by a presentation by Pascal van der Wele, and he will talk about the chimerism monitoring by NGS. And Pascal is a project manager R&D at GenDx.
Good day, ladies and gentlemen. My name is Pascal van der Wele, and I work as a project manager in the R&D team of GenDx. And today I would like to tell you something about how we plan to facilitate chimerism monitoring through NGS. For this, we are currently developing products called NGS Track and Track Engine. NGS Track is a package of reagents, and Track Engine is a software package for analysis. The reagents will allow you to go from DNA to sequencing in under 4 hours, with less than 2 hours of hands-on time, allowing for quick and easy workaround on the lab. For these products, a single pre-sample measurement is sufficient for all future monitoring experiments. So very little pre-sample is required for your experiments. And Track Engine software will allow for quick and easy data analysis. For these reagents, we are pursuing a sensitivity of at least 0.5%. And as a frame of reference, STR is between 1 and 5%, and qPCR is around 0.05%. So for NGS Track, we are working on 32 indel markers, which cover 362 nucleotide positions. So these markers are hypervariable and considered to be very informative for different samples that you might be testing. These markers are compatible with Chimer and the AlleleSeeker product lines, which are currently in our portfolio. The 32 different markers cover 18 unique chromosomes and also include one XY marker for gender determinations. So we've chosen for indels because they have greatly improved detection over single SNPs, meaning that you could detect an indel far lower at a far higher sensitivity than you would be able to detect a single SNP. This means that indels are less sensitive to sequencing errors, and you can clearly see this from the graph or the figure in the bottom right. So here we have an indel at 5%, which is clearly discriminatory from the noise. However, as soon as we move from 0. To 0.5%, you can see that the indel is about the same height of the highest noise levels. So if this were a single SNP, you would not be able to discriminate the SNP from the noise. However, considering this is an indel and this is a pattern that you're following, you can still clearly identify it at 0.5%. You can even identify it at levels as low as 0.05%, which means that indels can be detected below the noise levels. So the NGS-Track workflow consists of 4 independent steps, starting with amplification. So we amplify 32 markers in 7 different multiplex mixes. And during amplification, we perform indexing simultaneously. Following the amplification, we have a library preparation, consisting of a single pooled bead cleanup step, which is compatible with the NGS-Go workflow. Then we perform sequencing on your Illumina paired-end sequencing device. And finally, we have data analysis in Track Engine, which is a software package being developed in-house. And here on the bottom right, you can see the 7 different multiplex mixes that we have and the number of markers per mix. The estimated capacity on a MiSeq for genotyping experiments, meaning all 32 markers being tested, is between 32 and 64 samples for a standard flow cell, 8 to 16 samples for a micro flow cell, and 2 to 4 samples for a nano flow cell. These capacities depend on the sensitivity, and here a sensitivity of 0.5% is assumed. So monitoring experiments can actually be conducted with a subset of mixes, which increases the number of samples that could be loaded on a flow cell up to 3-fold, meaning that for a standard flow cell, you would be able to load around 100 samples at least, depending on the mixes that you choose. And all these reagents are compatible currently with paired-end sequencing on Illumina systems, and we will evaluate other platforms based on demand. So this is the NGS-Track proof of principle. And here you can see on the x-axis various percentages for sample 1, and on the y-axis percentages measured. So this is a dilution series of 2 DNAs in each other. So sample 1 is being diluted in sample 2. This is a theoretical measurement of the dilution series. And we are going to superimpose the actual measurements of the assay on top of them, which you can see over here. And you can see that the graphs clearly overlap, which means that measurements match the expected chimerism levels at both high and low levels of DNA. So following the proof of principle, we proceeded with proficiency studies. So we've participated in 5 proficiency studies, which were conducted between 2017 and 2020, and we tested these with NGS-Track. Each study consisted of 2 pre-samples, one for the recipient and one for the donor, and 2 post-transplant samples. On the x-axis, you can see the various samples that were tested, and on the y-axis, you can see the chimerism levels of the donor that were measured. So the acceptability bounds are marked in gray, which means that you should be with your measurements always between the gray lines. The black dots are the averages supplied by the proficiency studies that were participated in. And the blue dots show the averages that were measured with NGS-Track based on all the informative markers that were in the assay. So this means that NGS-Track passes criteria for all 5 proficiency studies. Then, parallel to reagents, we are also developing software, Track Engine, and we are basing this on our previous experience with NGS and qPCR for chimerism monitoring. So NGS Engine and Chimer Engine are being fused and used to create Track Engine. And here we have the splash screen on the right. Track Engine will present you with a dashboard for clear overview of all your results. It will allow you to import sequence data, to have a subject and sample management, to add subjects, to add samples, and it will present you with an overview of tasks that you might have open, as well as tasks that are currently being processed, which you can see over here. Track Engine will also contain a database for easy management of subjects and results. And considering databases can be quite large, we have included search and filter functionality for easy identification of your subjects and tracking them through time. And of course, we will allow you to perform detailed tracking of results per subject. We will show you trend analysis of your results, which you can see on the top left here, which can then be opened for a more detailed overview of all results. You can have an overview of all pre- and post-transplant samples tested, including their review status. We can have open tasks presented to you so that you always know what should be done, and all information of specific patients or subjects can be tracked in this view as well. So in summary, this means that NGS-Track and Track Engine are a highly efficient, time-saving alternative to STR. With a sensitivity around 0.5% or lower, this sensitivity will be far higher than with STR. We are currently developing 32 markers multiplexed in 7 mixes, and these markers are based on the well-established allele seeker and chimer product lines. Since indexing occurs during amplification, which minimizes hands-on time, the turnaround in the lab is very efficient and quick. And we will allow you to perform mixing and matching for monitoring experiments, so that you can have efficient use of your flow cell after your genotyping results. And then finally, Track Engine is being developed for quick and easy data analysis, going from DNA to monitoring very quickly. And with that, I'd like to thank you for your attention.
Thank you, Pascal, for this nice update on the chimerism monitoring by NGS. And now the last speaker will be Maarten Penning. He is Chief Technology Officer at GenDx, and he will update you on the product development at our company. Maarten, the floor is yours.
Hello, my name is Maarten Penning and I'm the Chief Technology Officer of GenDx. And I would like to thank the previous presenters, and I would like to discuss with you how their presentations will affect our plans in development. So I'd like to start with the chimerism monitoring products. We have already products for chimerism monitoring for many years. They're based on qPCR, and we also have a kit that can determine if a relapse is with HLA loss or without. The advantage of qPCR-based assays is that they are much more sensitive than the classical STR methods. With STRs, you can basically confirm a relapse, but with qPCR-based methods, it's really possible to detect recipient cells already before the relapse is occurring. Once you have noted there is a relapse, it's important to determine if it was a classical relapse or that it was a relapse with HLA loss, because that would require a different clinical follow-up. Both is possible with our qPCR-based assays. Now, as my colleague Pascal has presented, we are currently also developing a workflow for chimerism monitoring by next-generation sequencing. A product line in reagents called NGS-Track and the software Track Engine. As Pascal showed, it's a complete and user-friendly solution, very sensitive and very specific. Also very convenient, and many markers of which we think make a really useful and robust assay. What I would like to share with you is that the product is available, we expect the product to be available as of September of this year. The other project, as presented by CMAC before, is about MIG-A and MIG-B. Also for typing of MIG-A, MIG-B genes, we have a strategy available already for many years. It's based, like the HLA amplifications, on a full-length amplification of the genes. Typing you can do in the same workflow as the HLA, in the same platforms, and also in the same software, NGS engine. The important regions in MIG-A are the codon 129, demonstrated here. You can very nicely and specifically demonstrate, as well as the microsatellite region in exon 5. Also for MIG-B, these assays result in robust and reliable data. Now, we are expanding our portfolio in collaboration with a group of Professor Baram to also develop ELISA-based assays, both for MIG-A, MIG-B antibodies, as well as for MIG-A and MIG-B proteins. And for us, this is a really exciting new development, as it really is the first steps outside of the DNA typing field, but also supporting in the transplantation diagnostic laboratories. The hemato-oncology project was presented by Lisette van der Korput, and of course we also are working on these types of assays. As mentioned, in collaboration with the University Medical Center of Utrecht, which lab has developed these assays and is also using them in a routine. In the workflow, they combine the amplifications with the library prep of the HLA typing, and then run the assays in the same sequencing experiment. So as explained by Lisette, the goal of these assays is to subclassify all kinds of leukemias. Of course, we take care of the ELN guidelines and the WHO guidelines. And our goal in this project is to develop CE-marked kits. So this is an ongoing project, and we expect the first assays to be available around September of this year as well. One topic which was not discussed in this meeting before is the blood groups. Of course, we all know there's ABO, there's rhesus, but actually there's many more blood groups. About almost 40 different blood groups are currently described by ISBT. Typing of those blood groups typically is done by serology or by looking at a limited number of SNPs. So what we like to do and how we would like to support the blood group typing community is by providing whole gene sequencing assays for these rare blood groups, as well as support the curated database like the IMGT. This, of course, matches very nicely with the experience that we already have based on the HLA, MIC and CARE strategies that we are supplying to the field. So the current state of affairs is that we have developed assays for 8 blood groups. They vary in length, as you can see in the middle column. And the interesting thing is that if you test a very limited number of samples, 7 in this case, we find many new variants as displayed in the right column. Currently, we're also developing assays for the rest of the blood groups. So if there is a blood group that has your personal interest, please let us know and we can see what we have and if we could collaborate. Of course, we're also still improving and developing our HLA typing portfolio. So the latest addition and our new flagship product is our multiplex for 11 genes in 3 tubes, NGS-Go MX11 in 3. So why do we choose for 3 tubes for the 11 amplifications? That has to do with the fact that this results in the best data quality. So here we show a picture of HLA-B amplification in the singleplex amplification. You can clearly see nice 50-50 balance of the 2 alleles, well separated from the noise, which noise level of 1 to 2% is typical for Illumina sequencing. Now if you do multiplexing, in this example we have the 6 load C in 1 tube, you see that the noise is increasing. This is due to crossover events, hybrid reach which occur in your data. And if we would further multiplex by doing 11 genes in 1 tube, you see that the noise further increases. Now in our MX11 in 3 assay, you see that by doing separating the amplifications in 3 tubes instead of 1, the noise levels are almost as good as with the singleplex amplification, demonstrating that this multiplex is really high quality of data. Another big advantage of the MX11 in 3 multiplex is the increased coverage. So on the left I show the classical coverage of the MPX V2 products, and here on the right you see marked in these pink boxes that the coverage has increased for almost all of the class 2 genes. So this also offers a lot of additional data. So how does it work? Well, the setup is really simple. If you buy the kit, you get 3 tubes, all with the individual primer mixes, and 3 tubes of reconstitution buffer, a separate buffer for each of the mixes. You dilute reconstitute the primers, and of course there's long mix, the enzyme, and water that you need to set up your master mixes. So the 3 master mixes are identical, the cycling protocol is identical, and after cycling, amplification, you just pool the amplicons 1 to 1 to 1. So this slide summarizes the advantages of using a multiplex instead of the singleplex amplification. You see a lot less amplification reactions. You also see that you don't need to run a gel, and you don't need to quantify to normalize your pooling. You just pool 1 to 1 to 1 and then continue onto the sequencer. So in summary, MX11 in 3 is the assay with the largest coverage, but on top it offers the highest data quality. It has the largest coverage as said, and that as a result of that, it is the assay with the fewest ambiguities. Currently, we are wrapping up the CE registration of MX11 in 3, and once that's done, it shows that each step in the workflow can be performed with a CE-marked GenDx product. from amplification, library preparation, up to data analysis in NGS Engine. So I hope I showed you that we are evolving, we are developing new products, we are expanding our portfolio even beyond the HLA, while at the same time further developing and improving the HLA typing products that we already have. So with that being said, I thank you for your attention and I hope to see you soon.
So this concludes this session. I want to thank the speakers, Lisette, Siamak, Pascal and Maarten for their contribution. I'd like to thank you, our audience. If you have any question or like to have more information on one of the products or one of the presentations, please visit our virtual booth and you're able to chat with one of our colleagues. And by having said this, I would like to conclude this satellite symposium and I wish you a really nice remainder of the conference.